Living things often have a specific pH range that they can survive, and microbes known as acidophiles are capable of living in acidic environments, which are lethal to many other organisms. Why is a high level of acid dangerous to living things? Well, take for example the enzyme trypsin. It is a digestive enzyme, and the way that it can perform its function is by creating a pocket, an active site that has a specific shape, so that a reactant with a specific shape can fit inside. It does not work at an acidic pH, however. Other digestive enzymes, such as pepsin, they are the ones which work in the acid of the stomach. Trypsin functions in the small intestine where it is basic. Why? Protein structure is so complex, it involves a number of mechanisms, such as the formation of hydrogen bonds, which may contribute to the formation of alpha helices and beta sheets, and then the interaction between positive and negatively charged amino acids forming salt bridges, or also disulfide bonds, which can form a bridge. pH affects all of these. And so as one adds or removes more hydrogen ions, this affects the charges on the side chains of amino acids and thus alters which of these bonds that they can form. And so a protein's shape can change when the pH is changed because the bonds which are determining this structure, they change. It is thought that the early Earth was a very hostile place. Not only would it have been much hotter due to all of the volcanic and tectonic activity, but also this would have released a number of acids, such as sulfuric acid and nitric acid, coming from all of the elements released during the volcanic activity. As a result, it is not surprising that not only do eubacteria and archaea include both hyperthermophiles and acidophiles, that many of the hyperthermophiles are also acidophiles. They can live in both high temperatures and acid. And these cluster at the very root of the genetic trees of gene sequence comparisons. So in other words, it looks like the basal lineages of life on Earth, the most primitive organisms alive today, were adapted to both acidic and high temperature environments, given that is what we think was present in the early Earth. Although some acidophiles may share features which adapt them to acidic environments inherited from these early ancestors which lived in a primitive Earth. There are acidophiles which have adapted to acidic environments later. So protists like amoeba, ciliates, green algae, red algae, and diatoms can include acidophilic species, and as can fungi. And so therefore, some adaptations would be unique to each lineage. So some organisms would have adapted to acidic environments in different ways. So for example, one bacterium, Acidobacter aceti, is different from others. While most acidophiles maintain a neutral pH in their cytosol by, say, pumping out the hydrogen ions, Acidobacter allows the hydrogen ions to create an acidic environment inside the cells and have simply adapted the proteins of the cell to form normal structures in an acidic environment. There are other adaptations for acidic environments. For example, archaea possess different types of cell membranes with ether bonds with fatty acids which form a monolayer, and this not only helps protect them from high temperatures but also acid influx as well. Some amino acids are acidic, such as aspartic acid and glutamic acid. And in acidophiles, the frequency of the use of these amino acids increases, just as it does in the protein pepsin, which functions in the stomach. So it seems that changing the amino acids used in proteins can help adapt them to not change their shape in acidic environments. Acidophiles can have other ways of adapting to acidic environments as well. So, for example, in human cells, we have pumps which control the amount of potassium inside the cell, and thus the charge of the cytosol relative to the outside of the cell. Acidophiles often let in more potassium, which creates a more positive charge inside the cell, which reduces the amount of hydrogen ions which enter. Human cells and others typically have pumps which can pump hydrogen ions out of the cells. Acidophiles then could have a diversity of pumps and exchange mechanisms to get rid of the hydrogen ions which enter. Human blood and cells contains buffers which help to bind excess hydrogen ions, and acidophiles have these as well. Cells that can adapt to acidic environments not only have habitats where they can live with little competition, but also, just as human mitochondria make ATP energy by letting hydrogen ions go from an area of high concentration to low, these cells can let hydrogen enter the cell in a controlled way so that they can then generate ATP. And so hydrogen ions going from an area of high concentration to low is a potential energy source which can be harnessed.